so I'm going to go through all of the structures of the integumentary system that you need to be able to identify and know the function of. So when we're just looking at, so this is a section through the skin, two layers of the cutaneous membrane of the actual skin, and then we have a subcutaneous layer. So from here, the apical surface, to where we see the dermal papillae begin, this is going to be the epidermis, right? So the general layer here is the epidermis, made up of stratified squamous epithelium. Why stratified squamous epithelium? Remember, stratified squamous epithelium is found anywhere that's open to the outside, subject to abrasion. Cells can slough off, but we have lots of layers as backup, right? Okay. Then from here to here, where the adipose begins, this would be our dermis, primarily composed of dense, irregular connective tissue, dense collagen fibers oriented in all different directions, able to resist pulling forces in all different directions, but the fact that it's irregular means that it still has room for vascularization as well as other accessory structures being embedded within it, right? Then once you see the abundant adipose begin, we know that this is the hypodermis or the subcutaneous layer, primarily composed of loose areolar connective tissue, but abundant adipose. Why? Adipose adds cushion, so cushion between the outside environment as well as the muscles and other organs that lie below. That would be deeper to this, as well as adipose helps with thermoregulation and energy storage. These are all functions that we learned when we talked about tissues early on in the semester, right? So specific layers. In the epidermis, there are some specific layers that you should be able to identify. Now, apical surface, this would be the basement membrane separating epidermis from dermis. The very first layer, I like to start with the first layer being the deepest layer. This is the stratum basale. It's kind of, so if you focus in, so if you want to get a little bit closer, so this first layer of cells that look even kind of somewhat cuboidal-ish, the stratum basale cells, these are the cells that are constantly undergoing mitosis. Mitosis is just dividing, making more cells, right? Making more identical keratinocytes. So the stratum basale cells just produce more and more keratinocytes. Why? Well, as these cells at the apical surface slough off, we have to have more being generated, right? So here's the stratum basale. The next layer, so what I like to do, and I use this method when I'm identifying this on the microscope too, is the stratum basale layer is the easiest to identify because it's right next to the dermis, right? Then I go up and I look for the stratum granulosum, which is going to be this layer of cells that stains very darkly. It stains darkly because these keratinocytes are filled with keratin granules, which makes them stain very dark. So I look for the stratum basale, then I look for this granulosum. Everything in between, this is all stratum spinosum. So it's the thickest of all the layers, of all the strata within the epidermis. So if I put a sticker anywhere in here and ask for a specific layer, you would tell me stratum spinosum. What's the function? What's happening in the stratum spinosum? As the keratinocytes move up through the stratum spinosum, they get pushed up because what is the stratum basale doing? Always making more keratinocytes. So it pushes up the older keratinocytes, and as they get pushed up, they fill with keratin granules. They also start to produce a very hydrophobic substance, and they get coated in this hydrophobic substance, which is good because it helps waterproof our skin, but not good for cells trying to get nutrients, right? So as these keratinocytes fill with keratin and then coat themselves with hydrophobic substance, they start to die, okay? In the stratum granulosum, what's happening here is that these keratinocytes are super full with keratin, so much so that they really can't do anything else other than be full with keratin. So carrying out other metabolic processes are difficult because they're so full with keratin, yeah? And then after the granulosum, what happens is we have this layer, which I won't ask in a histology slide because it's difficult to see, but you can see it here on this model. This translucent layer right here is called the stratum lucidum. Stratum lucidum, the reason why it looks transparent or clear is because these keratinocytes have undergone apoptosis. So they've popped, and that's why it looks translucent, right? They undergo apoptosis for a couple of reasons. One is that they're super full with keratin granules, so they can't really do anything else metabolically. But they're also really far away from the vasculature of the dermis, right? Remember, epithelial cells are not vascular. Epithelial tissues are not vascularized. They have to rely on diffusion of nutrients. Where are those nutrients going to come from? From the blood vessels in the dermis, right? So as these cells move up through to the apical surface, they get further away from the blood supply of the dermis. So they don't get nutrients, they're super full with keratin, and then they die, right? They coat themselves with a hydrophobic substance, they fill with keratin, they don't get nutrients through diffusion, and then they undergo apoptosis. Okay, so that's the stratum lucidum. And then once they die, if we're talking about thick skin, these dead cells compact to form a stratum corneum. So this is this outer layer where we no longer see nuclei, big, thick, dead layer of keratinocytes. This is the stratum corneum. Over here, this would be like a representative of like thin skin. Well, it's just removed. If we were to just remove this layer, the stratum corneum, this would be thin skin. But because we have a prominent stratum corneum, this is thick skin, like the palms of your hands and the soles of your feet. Yeah? Okay, so it makes sense the stratum basale is the most metabolically active. They're undergoing mitosis all the time because they're closest to the nutrients. They can undergo mitosis very rapidly because they have lots of nutrients nearby. Yeah? Okay. If I were to draw an imaginary dotted line across here, it would divide the dermis into two specific layers. If I put a sticker in these structures right here, but I ask for the specific layer, you would tell me the papillary layer of the dermis. From here to here, if I put the sticker anywhere in the dermis down here, you would tell me reticular layer, right? Each of these structures is called a dermal papillae. What's the function of the dermal papillae? It helps increase the surface area so that more nutrients, more surface area means more diffusion of nutrients for the epidermal cells above. Yeah? All right, cool. All right. And then again, when you see prominent adipose, this is hypodermis. So some accessory structures that you should be able to identify within the integumentary system. Here, this is the hair shaft. It's a part of the hair that you can, like, hair shaft up here. If I put a sticker here and you tell me hair shaft, I'm totally fine with that. This is the part of the hair that you can touch and feel. Okay, it's made up of dead keratinocytes as well. I'm not going to ask the specific structures of like the hair follicle and the hair papillae, but if you're in lecture class, you may need to know those structures. Hair follicle is made up of the epidermal cells that surround the hair root and then the hair shaft up here. All right, let's talk about the accessory structures you need to know. Okay. All right, so this glandular tissue that looks like little grape clusters, this is a sebaceous gland. I know that because of the structure, the grape-like structure, and also because it's attached to a hair follicle. These are sebaceous glands attached to a hair follicle. They produce an oily substance called sebum. This is an example of an ecrine gland because its substance goes out to the outside. So sebum is an oily substance. It helps lubricate and moisturize the hair. It also ends up being released to the outside of the skin. So the sebum helps moisturize and lubricate the skin as well. We all know if our skin becomes too dry, it cracks. Cracked skin means you are open to bacterial infection. That's all bad. So sebaceous glands produce an oily sebum which helps moisturize and lubricate the skin attached to the hair follicle. Another structure that's attached to the hair follicle is this structure right here, right here, right here, erector pili muscle. It's little bands of smooth muscle, so it's under autonomic control. We can't make it happen. It happens automatically. When the erector pili muscle contracts, it causes goosebumps or your hair to stand on end. A couple of reasons for this. If you were to, if you think of lower mammals, I always use the example of a cat because I have cats. But when a cat, its hair poofs up and stands on end, it looks bigger or more intimidating. If a cat gets really cold, its hair poofs up and stands on end. It allows for air to get trapped closer to the skin. That air warms up and helps form like thermal insulation. It's also under sympathetic control, so when you are frightened or you are fearful, this erector pillae muscle can contract, and that's why you get goosebumps, like if you're scared. Okay, again, that whole like maybe looking a little more intimidating. Now, not so much important in humans, but it's stuck around through evolution. Okay, erector pillae muscle. Let's see. So let's talk about some nervous. Oh, another glandular tissue. Sorry. I talked about sebaceous glands. Another type of gland is an ecrine sweat gland. So it's a tubular gland, whereas the sebaceous glands have this grape-like cluster structure. Ecrine sweat glands are tubular. So if we took a cross-section through this, we would see simple cuboidal epithelium. Okay? It has a duct that leads to the outside. Ecrine sweat gland will produce a watery substance and a watery fluid, which when released to the outside, when we're really hot, if our body temperature increases past a certain point, ecrine sweat glands produce a watery sweat to the surface of the skin, which evaporates and cools our skin. So when the water evaporates off of our skin, it pulls heat with it, and so that's evaporative cooling. That's the function of ecrine sweat glands. This is an ecrine sweat gland as well. This would be the sweat pore. Ecrine sweat gland, sweat pore. Function, evaporative cooling. Let's talk about some nervous tissue structures. This guy up here, which is it's embedded in the dermal papillae. This is a Meissner's corpuscle. It is for light touch, so fine light touch. It makes sense because it's located closer to the surface of the skin, right? So it can discriminate light touch. Whereas these guys down here, which you can find deep within the dermis and even into the hypodermis, these are Pacinian corpuscles. These are deep touch receptors. So they are going to be able to sense deep pressure and vibrations. Makes sense, they're located deeper within the integumentary system in the dermis and then even into the hypodermis. So all of this has to depress to stimulate this, right? So it takes more pressure, deep touch. Meissner's corpuscles, light touch receptors. Pacinian corpuscles, deep touch receptors, yeah? And then up here, these nervous tissue structures, these sensory neurons that go up into the epidermis, these are called nerve endings. Oh my God, my brain is drawing a blank. Free nerve endings, there we go. Free nerve endings. On the free nerve endings, you can find receptors like thermoreceptors, pain receptors. That makes sense, right? If we touch a hot stove, we want those thermoreceptors to be more superficial, not all the way down here, right? Because if thermoreceptors were located all the way down here, then all of this would have to burn before we felt it. Yeah? But the fact that thermoreceptors are located up within the epidermis means that when we touch that hot stove, we can sense it immediately. Yeah? If something is cutting us and damaging our skin, we have pain receptors located on these free nerve endings that can tell us to get away from that painful stimulus faster. Yeah? Okay, cool. And am I forgetting anything? I think I got everything. Okay, all right, cool, that's it.